Hi everyone. So my name is Partho, and you all know. But so what we are going to today study? Today we are going to study a topic from class 11th biology chapter, that is biochemistry. So today our topic would be about enzymes. So if you are observing the thing what I have written on the board, that is Michaelis Menten equation, it belongs to class 11th biology, but that chapter is called biochemistry. But this equation is from enzymes. So before starting, we must know what is an enzyme. So enzymes are the biocatalyst that can increase or decrease the rate of reaction, and after the reaction it remains unchanged. That means enzymes breaks down something and it convert the substrate into product, something like that. So we can say that our equation is like E plus S, that is a reversible sign, equals to ES, that is enzyme substrate, and again it is converted into enzyme plus product. Okay, so we can say that the substrate has been converted into product, but the enzymes remains unchanged. This is the basic thing of enzymes. So I can also say that this is the entire reaction, as I what I said, that entire thing belongs to class 11th biology. 11th biology means we have to go a little higher. That entire reaction generally depends upon two factors. Number one factor is that ES concentration, ES concentration, that means enzyme substrate concentration, and number two is the uh, reaction reaction uh, reaction factors or rate of reactions. I will come to that point, don't worry. So these are the general thing. Okay, so that means if the medium contains that particular amount of enzyme and the particular amount of substrate, then it will form that enzyme substrate complex and the reaction will goes on, yes or no. And another thing that is important here, that the uh, some factors, I will not say that is a factors, that is k1, k minus 1, k2. Okay, so these are rate of reaction. Okay, so why I have given k1? K1 means in this rate of reaction the enzyme substrate will convert it into enzyme substrate complex, and this in this rate of reaction k2, that enzyme substrate complex will be converted into enzyme plus product. So um, enzymes are like very specific, and enzymes are protein in nature. Enzymes generally bind with a very specific substrate. Okay, so in this regards, I will suggest all of you that to go through that two models. That two models are lock and key model. Okay, allosteric models where enzymes is a lock and substrate is a key. After binding properly, it will convert it into product. There are several models. You can, all the students, you can reach out that things that are easy quite. Okay, and you know that is the IBN, international international mm, uh, nomenclature, something that I am not able to recall the name right now. So that have categorized the enzymes into 6 parts: oxidoreductase, hydrolase, transferase, isomerase, ligase, lyase. Okay, in your NCERT books you will find that. But what today's topic it is, today's topic, how to derive the formula of Michaelis Menten equation. That is very important for class 11th and for NEET also. So let me do those things. Okay, so what I can do here is that, what actually I can do here, that uh, the rate of formation of enzyme substrate complex is equals to the rate of dissociation of enzyme substrate. That means how much amount of enzyme substrate will form, it is equals to, it will break down. Okay, both are same. So I can write in this way that k1 into es equals to k minus 1 plus k2 es. Is it clear? This is the rate of formation and this is the rate of breakdown. Both are equal, yes or no? Now simple math, In simple math, we can say that ES by E by S equals to K minus 1 plus K2 by K1. Yes or no? I just put this value to this portion and this value to that portion. That's all. So K minus 1 plus K2 by K1, this entire thing is known as Km. I will let you know what is the meaning of Km. Okay. And what E and S, there is another thing we must go through this thing. I am writing this thing, don't worry. That total enzyme, that is E total. E total enzyme equals to free enzyme plus enzyme substrate complex. I guess this thing is quite new to all of you, but it is not that new. Let me explain all the things properly. Okay. So what I have written here, E total. E total means total enzymes, because here the main boss is enzyme, because all the formula is based upon the enzymes. So from here we can get that the what is the amount of free enzyme equals to E total minus of ES. Is it easy? Yes or no? So can I put the value of E to here? Yes or no? I can put the value here. So if I put the value here, then what I will get, let me rub the board little fast. The board is little uh, dirty right now. So what will be happened? So it was the meaning of Km. Hi Anna, boys and girls, this was the meaning of Km. So what would be there? Now I will say that Et minus of Es into S by Es equals to Km. Yes or no? I just put the value of this into here. Yes or no? The E means Et total enzyme minus of enzyme substrate complex. Yes or no? I have put the value. Now let me derive the formula little fast. Okay. Sorry for these interruptions, boys and girls, because I have to wash the board multiple times, as the board is not that bright. Sorry for this, but still we'll continue. So what I will say that now I am going to crisscross. Crisscross means I will multiply Km with Es. Yes or no? And I will multiply Et with S minus of Es with S, just basic multiplication. Now what I can do, I will write it like this, Km Es minus of, because Es is there. Yes or no? Km, now it will be plus Es S, and here would be Et S. Is it clear? Now I can common that Es, I can take the common Es. So what I will get, Km plus S equals to Et S. Simple thing, just I am putting this value this side, that value this side, that's all. Okay, now what I am getting, that Es means ETS by Km plus S. So now we are going towards that formula, because that formula says that that V0 equals to Vmax S by Km plus S. It is like quite similar na at that type of formula. So how that formula came? So we know that interreaction depended upon, at the beginning of the lecture, what I said, the enzyme substrate concentration and as well as the reaction rate of reaction. These are the two main factors by which that entire thing is dependent. So I can also write one thing here, that is V0, that is the starting point, okay, equals to K2 ES. You will say, sir, what is K2 ES? Think about it. In the beginning, what I have drawn, written in the board, E plus S, K1 reversible sign, K minus 1, that is ES, enzyme substrate concept complex, then K2, enzyme plus product. So this K2 is nothing but the uh, reaction, what that particular term now, rate of reaction or something like that. So this is the entire thing that will happen if the enzymes will properly are able to bind with the substrate. So now I will put, we have already get that value of ES equals to ETS by Km plus S. Now I have to just put the value. How I am going to put the value? V0 equals to K2 means E total S, E total S multiply by S. I know the board is little clumsy, but try to understand what I am saying in the class. And this is the Km plus S. Okay. Now tell me, this is the total amount of enzyme. Yes or no? That is acting with the substrate. If the total number of enzymes is acting with the substrate, that is the maximum potential of the enzyme. So what I can write here? That is the Vmax. That entire enzyme has reacted with the substrate. So easily I can derive that V0 equals to maximum potential of enzyme Vmax bracket में S by Km plus S. Go for the black side, what I have written from the blackboard. So this is the entire formula of Michaelis Menten. And you know, if I check that, if I put that V0, if I take the V0 half means if the value of V0 becomes half, so what will happen? Let assume that V0 equals to half of Vmax. Let assume half of Vmax. Then put it in the value. Then half of Vmax, sorry, my bad, half of Vmax equals to Vmax S by Km plus S. Yes or no? Now Vmax cancel. Now what I can get, that Km plus S equals to 2S. That 2 will go into that side, basic math na, and that we can say Km equals to 2S minus of S equals to S. So from this equation, if we consider as V0 equals to half Vmax, we can conclude that Km equals to S. That means Km is the Michaelis Menten constant, and it is that particular substrate concentration in which the maximum velocity of the enzyme becomes half. So let us recall all the things what we have learned. Today's session is that what is enzyme, how an enzyme works, what are the different types of enzymes, how to derive that Michaelis Menten equation, because Michaelis Menten equation is very important to know that in which concentration the enzyme is acting properly with the substrate. Okay, and what is the maximum capacity of an enzyme also. So these are the things. So we also able to know how this formula, that is V0 equals to Vmax S by Km plus S, how this formula came step by step. If anyone has any doubt, pause the video and also listen very carefully what I said, and for that and that portion, and also this graph is well known to all of you. The board is not that big, that is a little issue. So let me rub few things. I'm rubbing this one. Okay, so you have seen this diagram, yes or no? Hi Anna. Very easy diagram. This is the this x-axis means the substrate concentration, that y-axis means the efficiency of enzymes, yes or no? Hi Anna. And this particular point is called that Vmax. Aish. That particular point called the Vmax, yes or no? If I take, this is the half, that means half of Vmax, this should be the half of Vmax, then this intersect would be that Km, yes or no? Km means key. Km means it is that particular substrate concentration in which the velocity of the enzyme becomes half, and this is the thing that is mentioned in your class 11 book. So in this video we have learned the Michaelis-Menten equation, how to derive the equation, what is Km, what is Vmax. Okay, so hope this video should be very much informative to all of you, and stay tuned for the next update. Thank you. Bye bye.